After watching an hour or so of reality TV, you'd be forgiven for thinking that some people are only working at 10% brain capacity, but in actual reality, brain scans show that nearly every region of the brain lights up during even simple tasks, like walking and talking. Not only that, but the brain itself requires 20% of all the body's energy, and it would make little evolutionary sense to throw much energy away at something that's only minimally active. As animals, our capabilities have developed in part from our brain structures. We're actually able to trace our evolutionary history as we come to understand these structures. Less complex animals have simpler brains designed for basic functioning and survival, rest, breathe, eat, whereas more complex animals, like many mammals, possess brains that feel, remember, reason, and predict. These animals don't have all new systems. They have new brain systems built upon old brain systems. The brain is kind of like a set of Russian nesting dolls. The outermost wooden doll is the newest, most detailed, and most complex, but as you go deeper, the dolls become older and smaller and simpler and more generic. The innermost wooden doll is the oldest, most basic. It's like a fossil in your head. This inner core of the brain, sometimes called the old brain, still performs for us much as it did for our early evolutionary ancestors. It's anchored by the brainstem, the most ancient and central core of the brain where the spinal cord enters the skull. Above it, at the base of the skull, is the medulla. Here, old brain functions happen automatically without any conscious effort, the beating of hearts, the breathing of lungs, that sort of thing. The pons is perched on the medulla, and it helps coordinate movement. Above the pons, at the top of the brainstem, is the thalamus, a pair of egg-shaped structures that take in sensory information related to seeing, hearing, touching, and tasting. The reticular formation is a finger-shaped nerve network inside the brainstem that's essential for arousal, things like sleeping and walking and pain perception, other important functions. The baseball-sized cerebellum, or little brain, swells from the bottom of the brainstem and is responsible for nonverbal learning and memory, the perception of time, and modulating emotions. It controls voluntary movement, like your sweet dance moves, but it also gets impaired easily under the influence of alcohol, hence the term tipsy. So the old brain systems keep our body's basic functions running smoothly, the sort of stuff any animal might need. This is pretty much where the brain stops for reptiles. For higher functions, we look to the limbic system. This includes the amygdala, hypothalamus, and hippocampus, sort of a border region of the brain separating the old brain and the newer, higher cerebral areas. The amygdala consists of two lima bean-sized clusters of neurons and is responsible for memory consolidation as well as both our greatest fear and hottest aggression. Stimulate one area of the amygdala and a docile family dog suddenly morphs into a bloodthirsty Cujo. Shift that electrode over just a tiny bit and that dog will be cowering at butterfly shadow puppets. The hypothalamus keeps your whole body steady, regulating body temperature, circadian rhythms, and hunger. Also helps govern the endocrine system, especially the pituitary gland. You should also thank your hypothalamus for allowing you to feel pleasure and reward. Rats implanted with electrodes in the reward center of their hypothalamuses and given ways to self-stimulate those areas will essentially reward themselves until they collapse or die. So use with caution. The final part of the limbic system is the hippocampus, central to learning and memory, and if it's damaged, a person may lose their ability to retain new facts and memories. Now above all of this is the most advanced stuff, the stuff that you think of when you think of the brain, the gray matter. The two hemispheres of your cerebrum make up about 85% of your brain weight and oversee your ability to think, speak, and perceive. The left and right hemispheres govern and regulate differing functions, giving us a split brain connected by a structure called the corpus callosum. So, for instance, language production is controlled largely by the left hemisphere, while certain creative functions are controlled by the right. Though this has nothing to do with handedness or people having dominant sides of their brain being more analytical or creative or whatever. That's part of what we call pop psychology, a behavioral disorder in which journalists and armchair psychologists use research showing beautiful, detailed, intimately connected complexities of your brain to sell newspapers or reinforce previously held beliefs. Yes, some tasks are distributed to one side, but the sides are deeply and constantly connected. A statement as general as artistic people use their right brains is as useless as saying artistic people have particularly bumpy heads. Finally, covering the left and right hemispheres, we have the cerebral cortex, a thin layer of over 20 billion interconnected neurons. But let's not forget the unsung heroes of your nervous system, the billions of non-neuron glial cells, which provide a spiderweb of support that surround, insulate, and nourish these cerebral neurons. You've probably seen enough brain diagrams to know that the cerebral cortex's left and right sides are subdivided into 4 lobes: the frontal, parietal, occipital, and temporal, all separated by especially prominent folds or fissures. Each lobe does indeed have its own set of duties. The frontal lobes, just behind your forehead, are involved in speaking, planning, judging, abstract thinking, and aspects of personality. The parietal lobes receive and process your sense of touch and body position. At the back of your head, the occipital lobes receive information related to sight. And the temporal lobes, just above your ears, process sound, including speech comprehension. Remember that each hemisphere controls the opposite side of the body, so my left temporal lobe processes sounds heard through my right ear. And within these lobes, there are still more regions that have specialized functions. Your motor cortex at the rear of your frontal lobes, for example, controls voluntary movements and sends messages from the brain out to the body like pet that dog or pick up that mug, while your sensory cortex right behind it processes incoming sensations like, ooh, that doggy is soft, or god, that mug is hot. The rest of your gray matter is made up of association areas that are related to higher mental functions like remembering, thinking, learning, and speaking. But the thing about association areas is that unlike your sensory or motor cortex, you couldn't just poke one and create a neat response. Association areas are more subtle. They deal with things like interpreting and integrating sensory input and linking it up with memories. And they prevail throughout all 4 lobes, so brain damage to different areas will cause very different results. A lesion on a specific part of the temporal lobe may destroy a person's ability to recognize faces. Traumatic memories or overactive hormones can profoundly affect our behavior and emotions, all of which remind us how fundamentally biology and psychology are intertwined.
To begin, let's look at the primary function of the nervous system. The basic purpose is to coordinate all of the activities of the body. It enables the body to respond and adapt to changes that occur both inside and outside the body. Now, the nervous system is actually split into 2 parts: the central nervous system and the peripheral nervous system. We'll explore the peripheral later, but first, let's look at the central nervous system. The central nervous system is made up of 2 major structures: the brain and the spinal cord. As most people know, the brain is found within the cranium or skull, and there are 6 main sections among other structures within it. These 6 sections are the cerebrum, cerebellum, diencephalon, the midbrain, pons, and the medulla oblongata. The first section is the cerebrum. This is the largest section. It's divided into 2 major hemispheres, which are the right and left hemisphere. And the cerebrum is further divided into 4 lobes. These 4 lobes are the frontal, the parietal, the temporal, and the occipital. The frontal lobe is primarily responsible for reasoning and thought. The parietal is primarily responsible for integrating sensory information. The temporal is primarily responsible for processing auditory information from the ears. And the occipital is primarily responsible for processing visual information from the eyes. The second section of the brain is the cerebellum. This is the section located in the back of the head, below the cerebrum, and above the first cervical of the neck. It is responsible for muscle coordination, balance, posture, and muscle tone. The diencephalon section is found between the cerebrum and the midbrain. It contains 2 structures, the thalamus and the hypothalamus. The thalamus behaves much like a relay station and directs sensory impulses to the cerebrum. And the hypothalamus controls and regulates autonomic nervous system functions such as temperature, appetite, water balance, sleep, and blood vessel constriction and dilation. The hypothalamus also plays a role in the emotions such as anger, fear, pleasure, pain, and affection. The midbrain section is located below the cerebrum at the top of the brainstem. It is responsible for certain eye and auditory reflexes. The pons is located below the midbrain and in the brainstem. It is responsible for certain reflex actions such as chewing, tasting, and saliva production. And the last section is the medulla oblongata. It's the lowest part of the brainstem and it connects with the spinal cord and is responsible for regulating heart and blood vessel function, digestion, respiration, swallowing, coughing, sneezing, and blood pressure. It's also known as the center for respiration. Now that we've covered the brain, let's take a look at the other half of the central nervous system, the spinal cord. The spinal cord is the link between the brain and the nerves and the rest of the body. The spinal cord is divided into 4 different regions: the cervical, thoracic, lumbar, and the afferent and efferent spinal nerves, which merge to form the peripheral nerves. The afferent spinal nerves are responsible for carrying information from the body to the brain, and the efferent spinal nerves are responsible for carrying information from the brain to the body. Now, within this elaborate system of nerves, neurons, and dendrites, there is a system that regulates the functions of the central nervous system, which lie outside its major components, such as the brain and the spinal cord. This system is known as the peripheral nervous system and is subdivided into 2 smaller systems: the somatic system and the autonomic nervous system. The somatic nervous system is responsible for carrying motor and sensory information both to and from the central nervous system. This system is made up of nerves that connect to the skin, sensory organs, and all skeletal muscles. The somatic system is also responsible for nearly all voluntary muscle movements, as well as for processing sensory information that arrives via external stimuli, including hearing, touch, and sight. The structures that allow this communication to happen between the nerves throughout the body and the central nervous system are known as the afferent sensory neurons and the efferent motor neurons. Now, afferent simply means conducting inward, and efferent means conducting outward. So just like in the spinal nerves, the afferent neurons take information from the nerves to the central nervous system, and the efferent neurons take information from the central nervous system to the muscle fibers throughout the body. The autonomic nervous system is further divided into the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is vital to our survival. Have you ever heard of the fight or flight response to danger? The sympathetic nervous system revs up the body when confronted with imminent danger to either defend yourself or to escape the threat. The parasympathetic nervous system is the counterbalance to the sympathetic response to danger, whether real or imagined. Once the threat is gone, the parasympathetic brings all the systems of the body back to normal. The basic purpose of the nervous system is to coordinate all the activities of the body. It enables the body to respond and adapt to changes that occur both inside and outside the body. The 2 major parts to the nervous system are the central nervous system and the peripheral nervous system. The central nervous system is also divided into 2 major structures: the brain and the spinal cord. The brain is found within the skull or cranium, and it is made up of 6 main sections. These 6 sections are the cerebrum, cerebellum, diencephalon, the midbrain, pons, and the medulla oblongata. The other half of the central nervous system is the spinal cord, and the spinal cord is the link between the brain and the nerves and the rest of your body. The spinal cord is divided into 4 different regions: the cervical, thoracic, lumbar, and afferent and efferent spinal nerves, which merge to form the peripheral nerves. Now that we know the brain and spinal cord primarily make up the central nervous system, let's look at the peripheral nervous system. The peripheral nervous system is essentially the nervous system outside of the brain and spinal cord. The peripheral nervous system is then subdivided into 2 smaller systems called the somatic nervous system and the autonomic nervous system.
And the nervous system is divided into two main structural parts. The first is called the central nervous system, and the second is called the peripheral nervous system. Central and peripheral, and both of those are themselves divided into two main parts. The central nervous system is made up mostly of the brain, which is in the head, so I'll just color that in here in magenta, and the spinal cord, which is in the spine. So I'll just color that in blue. That's this long, thin, tube-like structure that goes down the spine. Now the brain is divided into a bunch of different parts, and I'll just mention some of the big ones for this kind of introduction talk. Here we're looking at the brain from the left side. All of this part on the top that has several different colors here is called the cerebrum. Cerebrum, which is the biggest part of the brain and the part on top. Now if we look down from the top, like in this drawing here, you can't see the parts underneath. You can only see the cerebrum. And you see that it's divided into a left and a right half. And we call those halves the cerebral or cerebral hemispheres. Cerebral hemispheres, like half a sphere. And there's a left and a right cerebral hemisphere. Now on this picture over here, we're actually looking at the middle part of the brain. So it's like we've cut down the brain this way and separated the left and the right cerebral hemisphere. So here we're looking at the left cerebral hemisphere, but we're looking at it from this direction. And we see that all of this stuff on the top is the cerebrum, or one of the cerebral hemispheres. And then the part on the bottom, which is smaller, we divide into two other parts. So first, let me just draw an outline around this part right here. And this is the part that hooks onto the spinal cord down here. And this part is called the brain stem. Let me write that out. Brain stem, which is all of this part right here that connects the cerebrum to the spinal cord that they've cut off down here on this picture. And then the brain stem itself is divided into three smaller parts. The very top part of the brain stem that connects to the cerebrum is called the midbrain. Midbrain. And the middle part that's just below the midbrain we call the pons. P-O-N-S for pons. Under the pons, and the part that actually connects to the spinal cord, is called the medulla, or medulla, or sometimes people use a longer name of medulla oblongata. And then the last part, but not the least part of the brain, is this big part in the back that's behind the brainstem and connected to the brainstem. And this we call the cerebellum. Cerebellum. Now sometimes brain structures are referred to by the names of the structures they developed from in the embryo. So here's a picture of a human embryo, and it's developing its brain here. And this very front part is called the forebrain, or it has a longer name of prosencephalon. This part behind the forebrain is called the midbrain, and it also has a longer name called the mesencephalon. And then the part behind the midbrain is called the hindbrain. Hindbrain, and it also has a longer name of the rhombencephalon. So the forebrain is going to become the cerebrum. The midbrain is going to become the midbrain, just this part of the brainstem up top. And then the hindbrain will become the rest of the brain, the pons, the medulla, and the cerebellum. So just in case you hear people referring to structures in the brain by these names, that's where those names come from. They're from the developing nervous system. Here's a drawing of the spinal cord, this kind of long tube that runs down the spine. And there's a number of structures coming out of the spinal cord that I'll talk about next. So those are the parts that make up most of the central nervous system. And everything that's not in the central nervous system we call the peripheral nervous system. And the central nervous system is called that because it's kind of in the center of the body. And then the peripheral nervous system is called that because it's going to go out all over the rest of the body. The peripheral nervous system consists of two types of structures. The first are called nerves. Let me just underline this nerve right here. These are these long stringy structures that are going to go all over the body. And nerves carry the axons of neurons. The second main structure of the peripheral nervous system are called ganglia. A ganglion is singular and ganglia is plural. And ganglia are these lumps that are attached to nerves, and they contain the somas of neurons. Now let me just draw that a little differently over on this picture of the spinal cord that has these nerves coming out of it. So here's one of these lumps, one of these ganglia that contains the somas of some of the neurons in the peripheral nervous system. And some of these axons traveling through these nerves are going to be carrying information in to the central nervous system from the periphery. So they're going to bring information in this way from out here in the periphery. And when they do that, we call those afferent neurons. Afferent neurons carry information in to the central nervous system. Now, other neurons are going to have axons that carry information in the opposite direction. So they're going to carry information away from the central nervous system out into the periphery. And neurons whose axons carry information away from the central nervous system, we call efferent neurons. Efferent neurons. Now, there are lots of these nerves that are going all over the body, and you can divide them up in a few different ways. But we usually start by dividing them into the cranial nerves, which are nerves that exit the skull or the cranium. So these nerves primarily come out of the brain, and they're passing through the skull on their way between the central nervous system and the periphery. Or the peripheral nerves can be spinal nerves, and they're coming out of the spinal cord and passing through the spine on their way between the central nervous system and the periphery. And I'll just draw a few of these, but there's actually lots of these. They're paired on both sides of the body, and there are 12 pairs of cranial nerves and 31 pairs of the spinal nerves. Now, if we go back to our drawings of the spinal cord here, we can see that the spinal nerves actually form from these two parts, which are called spinal nerve roots. And there's a root in the front, and there's a root in the back. And in this drawing, we're looking from the back. So here's the root in the back that has the ganglia. And the way these spinal nerve roots work are that the afferent neurons bringing information into the central nervous system travel through the spinal nerve roots in the back, and the efferent neurons that are carrying information away from the central nervous system travel in the spinal nerve roots in the front. And then they come together in the spinal nerves. So we call those mixed nerves, because they have a mix of afferent and efferent neurons usually. Now, as any of these nerves travel from their proximal origin, proximal just meaning close to the center of the body, toward their distal ends, the word distal just meaning far away from the center of the body, or you can think of the word distant, all the nerves are going to branch repeatedly. So they're going to branch, and then they'll branch, and then they'll branch again, and they'll just keep branching into tinier and tinier branches, because they have a long way to go and they have to spread all the way out through all the way through the body. And these proximal parts of the nerves are big nerves that we can see with the naked eye. But once you get to these distal nerves, after they've branched a bunch of times, they actually become microscopic. And they're little microscopic nerves that go all over the body, connecting the entire body back to the central nervous system. And this is true for almost all of the cranial and the spinal nerves. Oh, I almost forgot here. I have this other drawing to show the cranial nerves. So here's a drawing of the brain looking up from the bottom. And all of these long, stringy-looking things coming out of the brain are cranial nerves. They're going to pass through the skull on their way from the brain out into the periphery. I won't draw them all in here, but there are a bunch of these cranial nerves that are going to pass through the skull.
Neurons are the cells that make up our nervous system, and they're made up of three main parts. The dendrite, which are little branches off the neuron that receive signals from other neurons. The soma, or cell body, which has all the neuron's main organelles like the nucleus. And the axon, which is intermittently wrapped in fatty myelin. Those dendrites receive signals from other neurons via neurotransmitters, which, when they bind to receptors on the dendrite, act as a chemical signal. That binding opens ion channels that allow charged ions to flow in and out of the cell, converting the chemical signal into an electrical signal. Since a single neuron can have a ton of dendrites receiving input, if the combined effect of multiple dendrites changes the overall charge of the cell enough, Then it triggers an action potential, which is an electrical signal that races down the axon up to 100 meters per second, triggering the release of neurotransmitter on the other end, and further relaying the signal. So neurons use neurotransmitters as a signal to communicate with each other, but they use action potentials to propagate that signal within the cell. Some of these neurons can be very long, especially ones that go from the spinal cord to the toes. So the movement of this electrical signal within the cell is super important. But why does a cell have an electrical charge in the first place? Well, it's based on different concentrations of ions on the inside versus the outside of the cell. Generally speaking, there are more Na+ or sodium ions, Cl- or chloride ions, and Ca2+ or calcium ions on the outside of the cell, and more K+ or potassium ion, and A-, which we just use for negatively charged anions, on the inside of the cell. Overall, the distribution of these ions gives the cell a net negative charge of close to negative 65 millivolts relative to the outside environment. And this is the neuron's resting membrane potential. When a neurotransmitter binds to a receptor on the dendrite, a ligand-gated ion channel opens up to allow certain ions to flow in, depending on the channel. Ligand-gated literally means that the gate responds to a ligand, which in this case is a neurotransmitter. So let's take the example of a ligand-gated sodium ion channel, which when it opens lets sodium flow into the cell. The extra positive charge that flows in makes the cell less negative, since remember it's usually negative 65 millivolts, and therefore less polar. So that's why gaining positive charge is called depolarization. Neurotransmitters typically open various ligand-gated ion channels all at once, so ions like sodium and calcium might flow in, while other ions like potassium might flow out, which would actually mean some positive charge leaves the cell. In the end though, when it's all added up, if there's a net influx of positive charge, then it's called an excitatory postsynaptic potential, or EPSP. In contrast, the opening of only ligand-gated chloride ion channels would cause a net influx of negative charge, creating an inhibitory postsynaptic potential, or IPSP, making the cell potential more negative, or repolarizing it. Now, a single EPSP or IPSP causes only a small change on the resting membrane potential. But if there are enough EPSPs across multiple sites on the dendrites, then collectively they can push the membrane potential to a specific threshold value, typically around negative 55 millivolts, although this can vary depending on the tissue. When it hits this threshold value, it triggers the opening of voltage-gated sodium channels at the start of the axon, which is called the axon hillock. These voltage-gated channels open in response to a change in voltage, and when these open, sodium rushes into the cell. The influx of sodium ions and the resulting change in membrane potential causes nearby voltage-gated sodium channels to open as well, setting off this chain reaction that continues down the entire length of the axon, which ends up being our action potential. And when this happens, we say that the neuron has fired. Once a lot of sodium has rushed across the neuronal membrane, the cell actually becomes positively charged relative to the external environment, up to about positive 40 millivolts. This depolarization process ends when the sodium channel stops allowing sodium to flow into the cells, which is a process called inactivation. But this inactivated state's different from when the channel's closed, or open for that matter, which are the two states that most other channels have. The voltage-gated sodium channel, though, is unique in that it has what's known as the inactivation gate, which blocks sodium influx shortly after depolarization, and stays in this state until the cell repolarizes and the channel enters the closed state again, and the inactivation gate stops blocking influx. Even though that inactivation gate's not blocking it anymore, the channel's still closed, so no sodium enters the cell in this state. The middle open state's therefore the only state where sodium gets let into the cell through the channel, and this is a very short window of time. Now, in addition to these sodium voltage-gated channels, we've also got potassium voltage-gated channels, which are slow to respond and don't open until the sodium channels have already opened and become inactivated. The result of this is that after that initial sodium rush into the cell, potassium flows out of the cell down its own electrochemical gradient, removing some positive charge and blunting the effect of the sodium depolarization. These potassium channels don't have a separate inactivation gate, and therefore they stay open for slightly longer, which means that there's a period of time when there's net movement of positive ions out of the cell, which causes the membrane potential to become more negative, or repolarize. During this repolarization phase, the cell also relies on the sodium-potassium pump, which is an active transporter that moves 3 sodium ions out of the cell and 2 potassium ions into the cell. It's during this repolarization phase that the cell's in its absolute refractory period, since the sodium channels are inactivated and won't respond to any amount of stimuli. This absolute refractory period keeps the action potentials from happening too close together in time, and also keeps the action potential moving in one direction. The combined efforts of this pump and the extended opening of the potassium channels result in a small period of overcorrection, where the neuron becomes hyperpolarized relative to the resting potential. During this time, the sodium channels go back to their initial closed state, and for a short period, the potassium channels stay open. At this point, we're in the relative refractory period, since the sodium channels are closed, but they can be activated. Although because the potassium channels are still open and we're in a hyperpolarized state, it takes a stronger stimulus to do so. Finally, as the potassium channels close, the neuron returns to its resting membrane potential. Alright, as a quick graphical recap of this whole process, with membrane potential on the y and time on the x. First, we start at the resting membrane potential of around negative 65 millivolts, and voltage-gated sodium and potassium channels are closed. We then receive EPSPs enough to hit threshold at about negative 55 millivolts, causing voltage-gated sodium channels to open, and we reach a peak of about positive 40 millivolts, at which point the sodium channels become inactivated, and we're in the absolute refractory period. Voltage-gated potassium channels then open, and along with the sodium-potassium pump, start to repolarize the cell. So much so that it overshoots and hyperpolarizes the cell. Next, the sodium channels enter their closed resting state, while at the same time potassium channels start to close, meaning that we're in the relative refractory period. Until finally they all close and we get to our resting membrane potential once again. Alright, so this process of positive sodium ions moving in and depolarizing the cell transmits the electrical signal down the length of the axon. Great. But really this process isn't that fast. So that's where the fatty myelin comes in, which comes from glial cells like Schwann cells or oligodendrocytes. These myelinated areas don't have voltage-gated ion channels spanning the membrane, so ions can't simply flow into the cell. That only happens in the spots between the myelin, called nodes of Ranvier. So instead of propagating via channels, the charge essentially jumps from one node to the next. That said though, jumps isn't really the right term, and these ions aren't just diffusing down the length of the myelin to the other side. That'd be way too slow. What actually happens is more like the sodium ions rush in and bump other positive sodium ions already inside the cell, which bumps other ones, and so on until this wave of positivity reaches the next node. The charge moving in this way down the myelinated areas moves really fast, and is called saltatory conduction, which makes it look like the action potential jumps from one node to the next. Alright, as an extremely quick recap, neuron action potentials happen when dendrites receive enough EPSPs to open voltage-gated sodium channels, which causes rapid depolarization of the neuronal membrane and propagation of an electrical charge from node to node down the length of the axon.
In today's video, I'm going to talk about the autonomic nervous system. Students, look at this. Holy shit, this looks awful. We are going to do this Penguin Prof style. You want to stay tuned because we are going to cover a tremendous amount of stuff. So here's the overall view. We've got the periphery, we've got the peripheral nervous system, which brings information from the periphery into the brain and the spinal cord. This happens via a pathway we refer to as the afferent or sensory pathway. Within the brain and spinal cord, we have interneurons that lie entirely within the CNS. And then coming out from the CNS back to the periphery, we have what we call the motor or efferent pathway. That's actually what we are going to be looking at today. So there are two different categories of efferent pathways. We have the somatic motor neurons, and those innervate skeletal muscle, the muscle that you voluntarily move. The rest of your motor pathways are autonomic motor, and those innervate visceral effectors. And these are the ones that give students a really hard time, and those are the ones we're going to be focusing on. So the players in this story, the parts, if you will, we've got dendrites and cell bodies, and then emanating from those, we have axons. At the end of the axon, we have the axon terminal. The axon terminal releases molecules. We refer to these as neurocrines. Most commonly, they are neurotransmitters, but neurocrine is a more general term and will also include things like neurohormones, which are released into the blood. These molecules will be received by receptors on target tissues, and the transmission of a signal across a space like this, whether it's a synapse or through the blood, we refer to that as signal transduction. So we're going to be looking at the ligands, including acetylcholine. Now, when you see a neuron that produces acetylcholine, or you're looking at a receptor that acetylcholine binds to, we use the term cholinergic for that. So you'll see a cholinergic neuron, right? That just means the neuron releases acetylcholine. We also have the ligands epinephrine and norepinephrine, and those used to be called adrenaline and noradrenaline, and that's where we get this term adrenergic. Let's start with acetylcholine. So here's the actual structure, what it looks like, and we're going to look at the receptors that acetylcholine can bind to. The first are the nicotinic receptors. There are actually two subtypes. The only real difference is where they are in the body. But when acetylcholine binds to this nicotinic receptor, the response is always excitatory. So here's what happens. You see acetylcholine binding to this receptor, which is itself an ion channel. It allows sodium and potassium to move in the directions that they want to go. Now, sodium is going to flood in, potassium is going to sort of trickle out, and the effect is going to be excitation. Now, acetylcholine can bind to another group of receptors. These are called muscarinic receptors, and there are actually 5 subtypes of these. Some of them are excitatory, and some of them are inhibitory. So it depends on where you are in the body and which muscarinic receptor you're looking at. So for example, some of the inhibitory muscarinic receptors, you'll notice this is a G protein coupled receptor, and the effect is that potassium channels will be opened. This causes a hyperpolarization or an inhibition. And this is the kind of receptor that we see in myocardial cells. So this will cause a reduction of heart rate. On the other hand, in other tissues, acetylcholine will bind to other muscarinic receptors, for example, in the smooth muscle of the GI tract, and you get instead a depolarization. You notice it's still a G protein coupled receptor, but you get the closing of potassium channels. So you see, again, the meaning of the message depends on the ligand and the receptor type. So let's look at epinephrine and norepinephrine next. These are catecholamines, and there are 5 different receptors that these guys can bind to, and you'll notice that they vary depending upon where they are in the body. They vary in structure, and they also vary in sensitivity. So this column of sensitivity just means that, for example, the alpha-1s are more sensitive to norepinephrine than they are to epinephrine. For beta-1s, it's the same. Okay, but what happens when epinephrine or norepinephrine actually binds to these receptors? So when they bind to an alpha-1, what you get is an activation of phospholipase C. The increase in cytoplasmic calcium levels will also cause muscle contraction, things like that. If epinephrine or norepinephrine bind to alpha-2 receptors, you see a decrease in the second messenger cyclic AMP. And when epinephrine or norepinephrine binds to any of the beta receptors, you see an increase in cyclic AMP. So let's put it all together and go back to our little schematic of motor pathways. So somatic motor neurons release a neurotransmitter onto receptors on skeletal muscle, and the effect is always excitatory because the signal is always for contraction of skeletal muscle. Now, you might think that we could just take an autonomic motor neuron and do exactly the same thing. Unfortunately, no. The reason why the ANS can be such a pain is because it appears that it takes two neurons to do the job of one. You have neuron number one releasing a neurotransmitter not on a target, but on receptors that are far away from the target in what we call ganglia. In the ganglion, there's a neuron number two, which comes out and will release a neurotransmitter onto the final target. So in other words, you have two neurons in the autonomic motor division, while in the somatic motor, you have only one. That's confusing. We call these neurons not neurons number 1 and 2, but a preganglionic neuron and a postganglionic neuron. Hopefully that makes sense, right? Now, these ganglia, the location of them depends on which of the two divisions you are looking at. So sympathetic ganglia generally run right along the spinal cord, just parallel to the spinal cord. Most parasympathetic ganglia are out closer to the effectors. So the secret formula of the autonomic motor division is this. You got to know which neurocrine is released by the preganglionic neuron onto which receptor on the ganglion. Then you have which neurocrine of the postganglionic neuron and which receptor on the target. And when you know the combination of these four things, you got it, right? These are the only neurocrines we have, and these are the only receptor types you have to choose from. And that will be it. Let's look at the parasympathetic division. The preganglionic neuron in the parasympathetic division is always cholinergic, which means it releases acetylcholine. The receptors on the ganglion are always nicotinic. So that means this signal is always excitatory. All right, well, what about the postganglionic neuron? Oh my gosh, it's also cholinergic. That's cool. So it also releases acetylcholine. Now, the receptor type on the target cells, these are different. These are muscarinic receptors. So depending upon which type, remember there were 5 subtypes of those, you'll get either an excitatory or an inhibitory response. So for example, in the heart, it would be inhibitory, but in smooth muscle of the gut, it would be excitatory. And that, ladies and gentlemen, is the parasympathetic motor pathway. The sympathetic motor pathway, the preganglionic neuron, is also cholinergic. So do you see they're both cholinergic? They release acetylcholine. The receptors are the same. They're nicotinic as well. So the only difference between the two in this view is this postganglionic neuron. In the sympathetic division, it's adrenergic, and it releases the neurotransmitter norepinephrine. And the targets are going to be those adrenergic receptors, either the alpha or the beta. So again, the effect on the target will depend on which of the adrenergic receptors you've got. So here we are comparing the two. And by the way, I have drawn the lengths to be anatomically representative. So the parasympathetic division, that preganglionic neuron is long and the postganglionic neuron is short. The opposite is true in the sympathetic division, right? So that relates to where the ganglia actually are. Now, just for comparison, I'm throwing in the somatic motor division here. So you see the difference between the somatic motor, where you have only one motor neuron, and the autonomic motor neurons, where you have 2. Now, we're almost done. There's one more kind of weird one, and that's called the adrenal sympathetic pathway. So we have still a preganglionic neuron that is cholinergic, so it releases acetylcholine onto nicotinic receptors. But this isn't a ganglion. This is the adrenal medulla. So the adrenal gland is very strange because the cortex is actually a gland, but the medulla, the inner part, is actually modified nervous tissue. So it acts like a postganglionic neuron in a way, but it's not a neuron. So it's very strange, but it releases epinephrine and norepinephrine. It's actually, I think, 80% epinephrine, 20% norepinephrine. Looks like it may be releasing some dopamine as well, which is kind of exciting. But it releases those neurocrines. You see now why we call them neurocrines? Because they act like hormones. They're released into the blood, and they will bind to adrenergic receptors on targets all over the body. So that is a little bit strange. There is no postganglionic neuron in the adrenal sympathetic pathway. So now we compare all pathways together: somatic motor, parasympathetic, sympathetic, and the adrenal sympathetic. And ladies and gentlemen, I'm excited to tell you that that is it. Those are all the possible combinations. So if you can understand these pathways, you got it.
No matter what you're a fan of, you can probably think of two characters who fit the description. Holmes and Watson, Brennan and Booth, Kirk and Spock, Ron and Hermione. They're close, they understand each other in ways that others don't, and you really can't picture one without the other. But when stuff happens, they usually react in different ways. Like if you're snooping around Hogwarts at night and you come across some beastie, the smart money's on Hermione to come up with a spell that saves the day, while Ron probably standing there with his mouth open. And when Sherlock starts to go crazy because there hasn't been a fiendishly clever jewel heist or whatever for him to figure out, you can count on Watson to calm him down. Mostly. My brother John, who is a writer of books, will tell you that these characters are foils. They're not opposites exactly, they're contrasts, and the more you see them together, the more they seem to complement or round one another out. And that is basically your autonomic nervous system in a nutshell. Your autonomic system is the branch of your peripheral nervous system that regulates the functions of your internal organs, like your heart and stomach, and also controls your smooth and cardiac muscles, and your glands. All things that you do not consciously control, so yes, you could say it has a lot of power over you. But the funny, or maybe confusing thing about this system is that its effects on your organs and muscles and glands are by no means consistent. Not at all. At any given moment, whether you happen to be totally relaxed or completely flipping your wig, your autonomic system is constantly making involuntary, fine-tuned adjustments to your body based on what signals your central nervous system is picking up. This could mean changing your body temperature, sending extra blood to a particular area, slowing your heartbeat, or tweaking your stomach secretions. Its effects change depending on the situation you're in, and also which part of your autonomic system is in charge at that moment. Because this weird little corner of your nervous system that keeps you alive is actually run by two competing interests. Two divisions that serve the same organs, but they create opposite effects in them, battling it out back and forth to either excite your body's functions or subdue them. One of them is dedicated to amping you up and preparing you for activity, that's your sympathetic nervous system, and the other one talks you down and effectively undoes what its foil did, and that is the parasympathetic nervous system. Together, they are what make your body experience stress, fear, relaxation, and defiance, courage and cowardice, panic and peace. If there's an epic novel going on in your body right now, it is probably being written by these two. Let's talk about names for a minute. One of the two divisions of your autonomic nervous system is called the sympathetic system. That sounds kind of nice, doesn't it? It's like understanding and calming and telling you that it's not so bad after all. Wrong! Contrary to its comforting name, the sympathetic system is what sounds your internal alarm bells. It's the hardware behind the famous fight or flight response. It is synonymous with stress. If the sympathetic is for fight or flight, the parasympathetic is for resting and digesting. It's responsible for maintaining your body and conserving energy for later. I recognize that this is confusing. But when you explore the anatomy of these two systems, like we're going to do today, they start to make a little more sense. Because even though their basic components are essentially the same, their physical structures turn out to be different in a few really important ways. And those differences can help explain why they act like the foils that they are, and why sometimes you feel more like Sherlock than Watson, or the other way around. First big difference, the nerves of these two divisions originate at different sites in your body. Your sympathetic fibers are thoracolumbar, meaning that they originate from between your thoracic vertebra, where your ribs attach, and the lumbar vertebra, just inferior to your ribs. Early anatomists saw how a network of nerves radiating from the middle of the spine like this could quickly coordinate the functions of many major organs at once, so it was called the sympathetic system, from the Greek words for feeling together. But the nerve fibers of your parasympathetic system begin both above and below where the sympathetic ones do. They're craniosacral, meaning they sprout from the base of your brain, and also from your sacral spinal cord, just superior to the tailbone. And because the roots of these nerve fibers basically frame the starting points of the sympathetic nerves, they were called parasympathetic, literally beside the sympathetic. Another difference between these two foils? They're ganglia. Unlike your sensory or motor neurons, where a single axon can reach all the way from your spinal cord to whatever muscle or touch receptor it works with, both parts of your autonomic system require two neurons in order to work. And those two neurons meet in ganglia, clusters of neurons cell bodies that house millions of synapses. But where these ganglia appear relate to their function and which division of the autonomic system they're serving. Sympathetic ganglia are found closer to the spinal cord because in those fight-or-flight moments of high excitement or activity, they need to be able to send a single message far and wide, like the bat signal. This way, excitatory signals traveling into a ganglion near the spine, ganglion being the singular of ganglia, can trigger action potentials in a whole bunch of other neurons that lead to many different effectors, like the heart and lungs and stomach and adrenal glands. By contrast, most parasympathetic ganglia are found way out from the spine, near or even inside of their effector organs. Because this system is responsible for taking care of particular functions only when you have the time and energy to do it, like digesting food or excreting waste, it uses more specific strategic signals. It's more like Commissioner Gordon calling Batman on the bat phone one-on-one just to talk about how things are going and, whether Alfred's doing okay after his meningitis. It's private conversation. Not everybody needs to be involved. Anyway, because the ganglia of these two divisions appear in different places in your body, it also makes sense that their neurons themselves have slightly different forms, namely the length of their axons. Now, ganglia can be kind of complex. It actually comes from the Greek word for a knot in a string, so when dealing with neurons around these structures, we look at the fibers before they run into the ganglion as well as after they come out of it. Understandably enough, the axon lengths of the neurons before the ganglion are called the preganglionic fibers, and the ones coming out are postganglionic. The key here is that in the sympathetic system, the preganglionic fibers are much shorter than the postganglionic ones, which makes sense when you think about it because the sympathetic ganglia are really close to the spinal cord and the axons don't have or need very far to go from the central nervous system. But they do have a lot of distance to cover on the other side of the ganglion in order to reach their effectors, so naturally the fibers leading out of the ganglia are a lot longer. And foils being what they are, the reverse is of course true for the parasympathetic system. Since parasympathetic ganglia are so close to or even inside of their effector organs, the preganglionic fibers are a lot longer. They extend from the cranium and sacrum, where they start, out to the lungs or liver or bladder, wherever their effector is, where they reach their ganglion. From there, the postganglionic fibers are super short, just long enough to communicate with their effector. So once again, it's anatomy and physiology. The structure of each of these systems is related to its function. The sympathetic nervous system is set up in such a way that even a small stress signal sent down one path could trigger a response in many effectors at once, which is one reason why your reaction to a sudden stressful event can feel so all-encompassing. By the same token, the resting and digesting that's overseen by the parasympathetic system doesn't require urgent, all-hands-on-deck communication. If you need to process a burrito or take a nap or maybe a trip to the bathroom, it can communicate with the organs involved one-on-one. But still, none of this tells us how these systems do what they do, how these nerves communicate with your organs and muscles and glands in times of either stress or relaxation. So you're sound asleep when your smoke alarm goes off. Before you even know what's going on, you start to feel it. Those smoke alarms are loud for a good reason. Your heart starts to race, your breathing picks up, you become sweaty all over your body. You are stressed. And I'm not talking about the my iPhone just died kind of stress. I'm talking about the I'm afraid I might die kind of stress. Even though it's often seen as a dirty word, stress, like pain, isn't all bad. It's actually very useful if you're, trying to get out of a burning building. Your sympathetic nervous system is the part of your nervous system that responds to stress, and it does its job exceedingly well by focusing on what your body needs to do right now. Like when you're facing a life or death ordeal, you don't need to be digesting that cashew cluster in your intestines or producing reproductive cells or fighting off an infection. That's all stuff you can deal with later when you're out of harm's way, so your sympathetic nervous system sweeps these suddenly trivial functions aside to blast all of your energy to your brain and heart and muscles to deal with the threat at hand. So this is where I tell you that you're lucky to have a sympathetic nervous system and that it keeps you alive and that you would probably die in X period of time if you didn't have one. All of which is true. But here's the thing. The problem is, nowadays our body's stress responses are triggered all the time, practically every day, even when we are not in mortal danger. I mean, worrying about paying your wireless bill or being late for an important meeting, those things are terrible, but they will not kill you. But good luck explaining that to your nervous system, because your physiological responses to non-immediate stresses are largely the same as when you're fighting for survival. So if stress is like ruining your life, That's why. And that's part of the reason you should get to know how it works. Because by learning about your sympathetic nervous system, you come to understand one of the key players in the physiology of stress. You may recall from our tour of the anatomy of your autonomic nervous system that in both your sympathetic and parasympathetic divisions, almost every signal has to cross two synapses. Each neuron travels from its root in the spinal cord to a ganglion where it synapses, and yes, that is a verb as well, with another nerve fiber. And that one, in turn, leads to an effector organ where it synapses again to create whatever response was signaled, like sending more blood to your skeletal muscles or making your heart pump faster. But you gotta wonder, or at least I gotta wonder, how do these neurons and effectors actually communicate with each other, and how do all of those signals result in the high-octane sensations that we know as stress? By and large, the stress response includes two kinds of chemicals, both of which I'm sure you've heard of. The first, of course, are neurotransmitters. These are made and released from neurons themselves, and like we talked about in our lesson about synapses, they are what neurons use to communicate with each other or their effector organs across a synapse. The other chemicals involved in stress are hormones, which are secreted by your glands. There are at least 50 different hormones that work in your body right now, and they do everything from regulating your sleep cycles to making your body retain water so you're not dying of dehydration all over the place. I'm telling you all this now up front because hormones and neurotransmitters are 100% necessary for understanding how your sympathetic division ultimately works. But when you trace a single sympathetic signal from the initial stimulus to the final response, those chemicals can be kind of hard to keep track of. That's because the very same substance can have different effects, actually sometimes totally opposite effects depending on where it's received in your body. And to make things even more fun, even though neurotransmitters are part of your nervous system and hormones are products of your endocrine system, a compound can be considered either a neurotransmitter or a hormone even though it hasn't changed one iota depending on where it happens to be operating in your body. So all of this can make understanding your stress responses pretty confusing. You might even say, stressful. All right, we're going in. The smoke alarm wakes you up. You smell smoke. It is time to move muscles. Fast. Your brain sends action potentials down your spinal cord and preganglionic neuronal axons. Those signals flow all the way to their ganglia. When the signals reach the synapses inside the ganglia, the nerve fibers then release a neurotransmitter called acetylcholine, known to its friends as ACH. If you haven't heard of acetylcholine yet, you're gonna want to remember that name. In addition to working in sympathetic ganglia like this, it's also what the rest of your peripheral nervous system and lots of your central nervous system uses to communicate. So when it comes to nervous communication, ACH is really the coin of the realm, the premium currency. So that acetylcholine crosses the synapse, and if there's enough of it, it can stimulate action potentials in several neurons on the other end in the postganglionic fibers. That's all it does, but it's important. It's basically a signal booster. Those postganglionic neurons then carry the action potential to the effector organs, in this case, let's say, your leg muscles, which are gonna need an influx of blood if they're gonna hustle you out of that house. And at the end of that second postganglionic neuron, the fiber releases a different neurotransmitter. This one's called norepinephrine, and it is always norepinephrine that's released from postganglionic fibers in the sympathetic nervous system. It's what crosses that final synapse and creates a response in the effector, like opening up blood vessels that lead to the leg muscles. So the preganglionic fiber releases ACH, and the postganglionic releases norepinephrine. Boom. Congrats. Your life is on its way to being saved. But your body has more than one mechanism for responding to things, especially things like a burning house. There's another alternative for getting the message out. I mentioned those hormones, remember? In addition to nerve fibers that lead to ganglia and then your effectors, there's also a set leaving the spinal cord that goes directly to your adrenal glands. Like all preganglionic fibers, these release acetylcholine too. But here, the signal doesn't end up in another neuron that triggers blood vessels to open or whatever. Instead, it triggers your adrenal medulla to release a flood of epinephrine and norepinephrine hormones that rush through your bloodstream toward your heart and lungs and other organs. Now hold up, did you notice what I just said? Yeah, I said the adrenal glands release norepinephrine as a hormone. Whereas in that first scenario, I said that norepinephrine was a neurotransmitter that sent the final signal to control blood flow to the leg muscle. Now, how can I say both of those things? Because they're both true. Norepinephrine is both a neurotransmitter and a hormone, and which one it is depends on how it's being used. If it's being released from a neuron and traveling across a synapse, we refer to a messenger chemical, no matter what it is, as a neurotransmitter. If it's being secreted by a gland into the bloodstream for more widespread distribution, it's a hormone. Even if it's the same chemical. And to an effector, hormonal norepinephrine is just as good as neurotransmitter norepinephrine. But as scientists, we describe them differently differently because they're functioning differently. Now, the ways in which a neurotransmitter/hormone like norepinephrine works is a good example of another confusing aspect of your sympathetic nervous system, because it works by both stimulating and inhibiting the same systems in your body at the same time. So, in our house burning scenario, the norepinephrine your system releases causes an increase of blood flow in some parts of your body, like your leg muscles, while restricting blood flow in other places where it's not urgently needed, like your guts. How can the same chemical cause opposite responses? Well, it all depends on the particular kind of receptors that an effector has for receiving that chemical. In the case of norepinephrine, its effector is smooth muscle, the muscle that controls all of your involuntary functions of hollow organs like the stomach and bladder, and also your blood vessels. On the smooth muscle cells controlling some blood vessels, there are receptors called alpha receptors. When norepinephrine or epinephrine bind to those receptors, they make those smooth muscle cells contract, thereby restricting blood flow. But on smooth muscle cells that control other blood vessels, there are lots of beta receptors for epinephrine and norepinephrine, and when they are activated, they make the muscles relax, letting more blood flow through. So it makes sense that the smooth muscle around your blood vessels, which feed your skeletal muscles, which you'll need to get out of that smoky house, are covered in beta receptors, because you want those blood vessels to relax and provide plenty of oxygen to the muscles in your arms and legs. And since running away is more important than digesting your dinner, the blood vessels leading to your stomach and intestines have lots of alpha receptors, which reduce blood flow to those areas because that burrito can wait until you're out of the house. So there's a lot going on in your sympathetic responses, and much of it can seem complicated or even contradictory. But the thing is, all of these functions work together to create a full body response, which is exactly what you need in an emergency. After all, it wouldn't do you much good to speed up your heart without sending that blood to your muscles where it's needed. It's up to those neurotransmitters and hormones and the receptors on the corresponding effectors to make sure that everyone is on the same page. So, the system works well, really well, sometimes too well. Remember when I said at the beginning how your body doesn't know life-threatening stress from life-annoying stress? Since your body's reaction tends to be a full-body response either way, it can become pretty taxing over time. I mean, we're talking about throwing parts of your body into overdrive while depriving others of blood and oxygen. That's not something you want happening every morning. So the irony here, the real kick in the head, it's that non-life-threatening stressors can actually end up endangering your life in the long run because your body's stress response is so effective. The frequent activation of your sympathetic nervous system and the triggering of the other part of your stress response, the part that's driven by hormones, can have nasty consequences like high blood pressure, digestive problems, and even the suppression of your immune system. So what your body needs to do is figure out how to relax, rest and digest. Feed and breed. That is where your sympathetic system's more mellow half-brother, the parasympathetic system, comes in. Consider your heart for a moment. For the average person at rest, like you probably are sitting there watching me, the heart beats at around 60 beats per minute. Once a second, nice and easy. But if you were to somehow disconnect your heart from your autonomic nervous system, things, as you might imagine, would change. But your heart would not stop. Actually, it would be the opposite. It would speed up. It would start beating at around 100 beats per minute, and that's just at rest. With your heart beating 2/3 faster than normal before you even broke a sweat, your cardiac muscle would experience a lot of extra wear and tear, the surrounding blood vessels would be under enormous pressure, and your body would suddenly require and waste a lot of energy. Basically, you'd be out of balance. Part of what keeps your heart under control is your parasympathetic nervous system. It's often described as the calming side of your autonomic system, a kind of antidote to the effects of stress created by the sympathetic system. But it's really much more than that. Unlike your sympathetic division, which lets you deal with the crisis of the now, the parasympathetic system allows your body to handle everything else. It not only calms you down after being stressed out, it's what allows you to digest food, to reproduce, to excrete waste, to fight off infections. Basically, it lets you do the business of living. But our bodies can only do that when they are in balance somewhere between excitement and inhibition, both aroused enough and calm enough to keep things working. So the parasympathetic system is why our hearts don't pump so hard that they explode, sure, but it also explains a lot of other stuff about our bodies. But just one thing. Learning about the parasympathetic system is going to involve a lot of memory. memorizing. Hope that doesn't stress you out. You will recall that our sympathetic and parasympathetic systems not only have different functions, more or less engaging the same organs to opposite effects, they also have different structures. Their ganglia, for example, are located in different places. The sympathetic ganglia are located near the spinal cord, while on the parasympathetic side, they're close to the effectors. And likewise, the use of neurotransmitters in the two systems is similar, but not quite the same. In both systems, neurons release acetylcholine, or ACH, in their preganglionic synapses. But in your parasympathetic system, the postganglionic neurons release ACH at their synapses with the effector organs too, as opposed to in the sympathetic system where the effectors get a dose of norepinephrine instead. But the biggest anatomical difference between these two systems has to do with the physical networks that they form as they reach throughout your body. While the sympathetic nerves all spring from the thoracolumbar area of your spinal cord, right around your midsection, the nerves of the parasympathetic division are craniosacral. And with the exception of a couple of sacral nerves near the tailbone that run to the bladder and genitals and rectum, most of these nerves never go through the spinal cord. Instead, they run right from the brain, almost all the way to their effectors. There are 12 of these cranial nerves, and they vary in terms of what kinds of neurons they contain. I mean, we're talking about the autonomic system here, but they are not all autonomic motor fibers. Some of your cranial nerves also carry motor fibers that control voluntary functions like moving your eyeballs around, and others carry only sensory fibers which relay data to and from your sensory organs. And, just to keep things interesting, some of your cranial nerves carry both motor and sensory neurons. So, which ones are where, and what exactly does each one of these 12 nerves do? As anatomists, we have to keep track of the human wiring diagram that are cranial nerves, because you don't want to end up like some sidekick in a 90s action movie who has to defuse a bomb all by himself. Should I cut the red wire or the black wire? Honestly though, if you find yourself inside of somebody's brainstem, you probably shouldn't be cutting anything. Since all 12 of these cranial nerves are important, you're gonna have to come up with some kind of mnemonics to help you keep track of both their names and their functions. You'll need to know what each one is called, whether it's a sensory nerve, a motor nerve, or both. And the map that we follow of the cranial nerves is based on a ventral view of the brain, looking at its underside, with the anterior portion at the top and the posterior on the bottom. First, let's tackle the names. Starting at the top, the first cranial nerve you encounter is the olfactory nerve, which takes scent information gathered by the nose and sends it to the brain, followed by the optic nerve, which does the same, but with visual data. Then there's the oculomotor, which controls 4 of the 6 muscles that control the movements of your eyes. The next nerve near the center of the brain's ventral side is the trochlear nerve, which controls a single muscle in the eye, and it lets you do this. Just below that is the trigeminal nerve, the largest of the cranial nerves, which branches into 3 main strands, hence the tri, and innervates the face and jaw muscles. After that, there's the abducens, which stimulates the muscles that let your eyes do this from side to side, followed by the facial nerve, which operates the muscles that make most facial expressions possible. Then there's the auditory nerve. You could probably guess what that's for. You might notice that up until the auditory nerve, the cranial nerves mostly control organs in the front of the cranium, mainly the eyes and facial muscles. But as you work your way down, the nerves tend to innervate the lower and more posterior portions of the head, like the glossopharyngeal nerve, which leads to your tongue and your pharynx. That's followed by your vagus nerve. You should definitely remember that one. And then the spinal accessory nerve, which has to do with moving your head and shoulders, and not whether your belt matches your shoes. Lastly, there's the hypoglossal, the nerve that allows you to swallow and talk, among other things that you do with your mouth and tongue. That was a lot of information and probably new words, so how are you gonna remember it all? Well, by finding a way to remember the first letter of each name in order, which is OOO-TT-AFA GVSH. That doesn't spell anything useful at all. There is a mnemonic that you'll probably hear in school that goes like this.
On old Olympus towering top, a Finn and German viewed some hops.
That's pretty weird sounding. Not terribly easy to remember. I mean, Olympus? Finn? Hops? There's gotta be something more relevant to us 21st century science lovers, like the Lord of the Rings fans out there might prefer something along the lines of
Onward, old orcs, toward the Argonath for a great villain. Slay hobbits!
I'm just trying to help. Whatever device you use to remember the names of the cranial nerves, you also have to keep track of their functions. That is, whether they're sensory, motor, or both. So again, from top to bottom, a lot of teachers use this sequence of S's, M's, and B's to remember.
Some say marry money, but my brother says big brains matter more.
That's not so bad, but I don't know, maybe you'll have better luck with something like this.
Sorry, Sherlock, mean Moriarty beat me, but some bobbies busted Moriarty masterfully.
You are, of course, invited everybody to think up your own, and feel free to share them in the comments. Hopefully there'll be some good ones down there. Anything would be better than fins and hops. But if you're gonna commit one cranial nerve type to memory, it should be 10, the vagus nerve. This long and extensive nerve stretches from near the brainstem down to most of your visceral organs, including your heart, lungs, and stomach. The vagus nerves work as a two-way street, ferrying incoming sensory information from the peripheral system to the brain and transmitting outgoing motor instructions from the brain to the rest of the body. So it's a B nerve because it has both sensory and motor functions, and usually you don't notice this nerve at work because its functions are mostly automatic. So you've had a really stressful day, so your sympathetic system is charged up. You come home, crash on the couch, mow down a half a pizza. Your stomach sends signals to your brain through the sensory nerve axons in your vagus nerve, telling you that your belly is full of starch and protein and fat. Your brain sees that your stomach is churning away, which is a usual parasympathetic activity, so it sends signals back down through the vagus nerve, triggering other parasympathetic responses, like slowing down your heart rate, putting some glucose back into storage, and reducing all that norepinephrine that your sympathetic system was pumping out all day. Soon, you start feeling more relaxed, which is just one reason why for some people, eating is a way of reducing stress and anxiety. In fact, it can feel so good that even though your stomach is full, you might continue eating. So, like I mentioned before, it can be easy to think of the two divisions of your autonomic system as opposites or even rivals, but that's a little off the mark. Looking at your body as a whole, you should picture them as two sides of a scale. Sometimes it's balanced in the middle, and sometimes it leans to the left or right depending on what's happening. That balance is the essence of homeostasis, and as you'll recall, homeostasis is the key to life. Here's something else that's important for life. Sex. It mostly falls within the parasympathetic domain of necessary, but not an emergency. But in order to effectively do it, you need help for both systems. First, the parasympathetic system has to make sure you're calm enough to even think about sex, and then funnel extra blood away from your muscles and down to your genitals, which is why too much stress and anxiety can lead to sexual dysfunction. But you also need a burst of that sympathetic system to excite you and keep you excited. So like the two sides of the scale, the balance depends on having the right amount of both. The rate of action potentials traveling through each division is known as your sympathetic tone and your parasympathetic tone. And most of the time, our parasympathetic tone is actually dominant, keeping down the caged animal that is your sympathetic response. That's why you need your parasympathetic system to keep your heart from racing like a rabbit's, and why most of the time our bodies can do the eating and sex having and all the other fun tasks that make up the business of living.